We're early day one, so it's been basically the first roundtables have gotten started, and always a great meeting. ISCT global event with everybody here, so kind of reconnecting with colleagues. I would say for me the big themes that I've seen almost every roundtable touch on thus far are automation and digitization. Essentially, how do we as a field take advantage of these technologies that have been around in other fields for a long time? There was a good panel just about an hour ago talking about just that and making some comments that the strides both automation and digitization have made in the last handful of years essentially dictate and should dictate the entire field to reevaluate what we think is a standard way to develop research and then drive to clinical and commercial manufacturing. Basically, early on, the technology wasn't really there for us to drive efficiencies, and it was more science project. And now that we have proven tools and technologies in both realms, basically, how do we as a field implement those in a standard way? And the question that the panelists were bringing up around that, because of course there's challenges implementing both. It's a little more complex than manual, but how many patients do you want to treat? That's how people should be approaching that. Therapeutic developers, GMP facilities around the world. If you want to treat more patients, you have to adopt these pieces of technology and these strategies that are going to empower you to actually get these out and not be stuck to a few centers globally. We need them to be essentially democratized. And maybe one final theme, Jamie, that came up quite a bit was this idea of data validation and process validation still being a difficult thing for companies to grapple with. And a focus point was a few of the panelists saying, and they're from the CDMO background, saying they like all the new tools and shiny gadgets coming out that can do everything all the way down to single cell analysis. But if those pieces of equipment arrive at these labs without a strong validation package that these companies and manufacturing organizations can quickly implement, they essentially have to invent their own validation and invent their own process to validate around. And that was a big ask from all these panelists as and I represent a tool and technology provider. So it struck home, and that is our strategy. How can we create simple tools that are easy to validate? There's a few. I'll touch on maybe the top 4 that come to mind, but back to automation. Where and when do you implement automation? That's a key. How do we drive consistency and repeatability within an autologous cell therapy manufacturing modality? Again, patient-specific, each batch is unique. So how do you ensure repeatability and scalability there? And then can allogeneic cell therapies, can we as a field make those in a way that have the same potency and clinical benefit that the autologous cell therapies have proven clinically for more than a decade and now commercially for quite some time? I think the jury is still out to make sure that those cells are potent, tolerated, and that we have the ability to have long, durable responses. And then finally, talking about automation and digitization, how do you implement these new technologies while also remaining flexible so that you can modify and improve your manufacturing process? Because these are long life cycles to develop a process or develop a drug and commercially launch it. You're talking at best maybe 6-7 years, probably more like a decade. So you can't start from a mindset where you're going to lock yourself into an inflexible strategy. You have to be able to adapt and incorporate new tech. I'll say one thing first. ScaleReady and our tools and my background with a cell culture bioreactor that grows T cells and NK cells better than anything in the world. It's called G-Rex. My area of expertise is certainly cell therapy, but I'll paint with a broad brush for a moment and talk there and how in general you could describe a lot of the manufacturing of gene therapies as manufacturing of virus as an example. And in that regard, you can lean on the traditional background of bioprocessing like monoclonal antibodies. Ultimately, the product that is generated during these manufacturing processes is not the cell themselves. So you're less concerned with the health, viability, and potency and consistency of the cells that are being produced, because you're actually using the cell like a factory to generate the product that you want. Within cell therapy manufacturing, it's flipped, right? The cell is the therapeutic agent. So you have to be focused on quality of the cell, potency of the cell, and consistency of the cells throughout the manufacturing process. So I do think that it's a completely different world, whether you're managing, that traditional kind of bioprocess mentality within gene therapy manufacturing, or cell therapy is different. I'll make one note, there's an in-between, which is gene modified cell therapy. So if you're gonna genetically modify a T cell or an NK cell, as is the case with a CAR T or a CAR NK, you are introducing, well, first off, you do that so that you can have potentially better targeted cells towards the cancer or disease, and also a more potent cell. But there's a trade-off, because when you genetically modify a cell, the regulatory agency is the level of scrutiny and the requirement for patient follow-up goes much higher. So I think that's something to consider. And finally, I would just talk about weighing those benefits of potential improved potency and targeting when you're genetically modifying cell therapy versus the added risk and cost when you're genetically modifying. So could you actually utilize native T cell receptors where you don't have to genetically modify? I mean, honestly, when you talk about cost, it has to be the focus. It's going to always be the focus at these conferences. We are getting durable responses, complete remissions. In cancer treatment, we never say the word cure. That's as close as we'll come. But we're 10 years, 11 years, 12 years out of some of these first patients who have no discernible cancer any longer, because those cells are persisting in the body. But even with a cure or close to it in cancer treatment, $500,000 US or more is really not a sustainable model for any healthcare system around the world. So if they remain that costly, patient access will continue to be diminished and only available to a select few in certain populous parts of the world. So that has to be the focus. Now, thankfully, how can we get there? How can we drive costs down? For me, it's relatively straightforward. The focus has to be simplification, simplify your process, get to know how you're manufacturing these cells, and basically strip away any unnecessary complexities that simply add cost and honestly add risk, because if it's overly complex, a lot more can go wrong. But from simplify, and one of the things I'll like to talk about is how do you then standardize as an industry? I think it becomes a lot easier to standardize and agree on best practices if you start from simple approaches that work and have been proven effective and kind of gotten the blessing of the regulatory agencies. Now we can, as an industry, align on standard manufacturing approaches, and then you automate essentially what is left. And tying that back together, for me, it's simplify, standardize, then automate, because we don't want to be automating a complexity into a manufacturing process. That's where your cost comes into play, and you can't drive costs down any further. So that's the big driver here in how to bring costs down, is we must simplify. And when I say simplify, I'm basically saying, can you make the process so simple that it's automatic, or at least components of the process? Because we want to make it automatic, standardize, and then we'll use technology, automation, robotics to automate the last pieces that still require intervention. Well, there's a lot of exciting potential here, but for me, it's back to that standardization approach. So both in terms of the technology itself, can we align on tools that are working and work in certain cell types and certain modalities, but also in the strategy people are using to manufacture. That's a lot to ask right now, because there's so many avenues and so many choices that developers need to make and go down. But I think if we force the issue on simplification, and we do that in a way that ensures the different disparate pieces of technology that are either available today or are being developed or will be developed, if we focus on making them simple but also flexible, where they can be connected to one another, think of like a Lego set, right? You can connect different pieces. That ability to make them friendly to that connection, both in terms of like physical connection and then in terms of the digital piece, in terms of getting that data out, I think that's what I'm most excited about. Myself being in the tool provider realm, and a lot of close colleagues and people I respect in the field, that is where people are focusing on, is like, how can we outwardly share best practices? And I think what it enables when we do that are two things. One, speed of development. We're going to be able to develop drugs faster because people aren't reinventing the wheel to solve the same challenges. And then when we do that, we actually allow, and this maybe is a little counterintuitive, you're saying simplify, standardize, and I'm saying it's going to speed up the development. It's actually going to allow us to make process changes faster and more effectively, because if we have standardized approaches and you have a well-characterized manufacturing process and well-characterized cells, if a new piece of technology comes along that you want to fold in and maybe swap out a module, it's easier to do that with that approach. And again, these manufacturing changes, which are really continuous improvement, which is what every manufacturer should strive for, they're almost impossible, and they're so scary internally because people are worried about what the regulatory agencies are going to see that as, or if they can link back to that earlier clinical data. But you can do that when we actually have aligned on approaches and we're sharing best practices in the field. Yeah, as far as future kind of forward-looking prospects, yeah, great question. I mean, there's a lot of exciting stuff, especially being at a conference like ISCT, which wraps academia, process development, and clinical, and the commercial end of things. The most exciting thing for me, having been around this space for 10, 11 years now, is I think we're getting back to our roots as an industry and becoming more scientific in our collaboration. And to turn a negative into a positive and basically see opportunity, the financial headwinds, the inability to raise capital when capital was so easy to raise for the last several years, it's actually forcing us to think more scientifically and say, can we collaborate more openly? Can we share best practices? Can we stop holding, every single piece of our process as, our differentiator? Can we actually share that as a group? Because it's more thinking altruistically instead of holding on to one thing as an individual entity, company, lab. If you share that and then the next lab shares that and so on and so forth, the speed at which we can improve our understanding and knowledge within the space is going to increase dramatically. And again, everybody to a person at this conference and everyone within cell and gene therapy is focused on how can we treat more patients? How can we get these therapies to more parts of the globe that are currently underserved? And again, I think getting back to the scientific roots is what I feel there's a big momentum swing in right now, and I'm super excited about it.